this or something that might just be the head of this, which would be known as like it's an icosahedral shape. That's what we typically think of. If you are somebody who studies viruses for a living, there's actually a distinction. This is what we would call the viral particle or the virion. The virus is once the genomic material has actually gotten into the host cell. So that's just a small technical piece of it. I always tell everybody that this always reminds me of a great Disney Pixar movie. What movie might this remind you of? Yeah, right? So, somebody watched the lecture. Well, huzzah. So, we've got our tails, we've got the head. What else do we have in this structure? So, here's our head, or we could call this actually the capsid, which is a more specific name. What is here? What's in this head?
Genetic material.
Yeah. Bacteriophages, it's usually DNA. Usually DNA. 96% of the ones that have been identified have DNA as their genetic material. That's not necessarily true for eukaryotic viruses. So when we get back from break, one of the things that you're going to see is we're going to talk about 3 different classes of viruses and very, very broad overview strokes. But in that description, you'll see some actually have RNA genomes. This is not something that we see in prokaryotes. It's not something we see in eukaryotes. It is something that's unique to viruses. So genomic material is located here. Okay. What else do we have? What are these little things? Tail fibers. What's their role? Attached to a cell. And we'll talk about that in a minute. But the attachment is actually the first step of any viral infection. Okay? These tail fibers define what we call the tropism, or help to define which cells will this particular bacteriophage be able to infect. Okay. What else do we have? What is this structure here? That would be the sheath. Won't really worry so much about what that does right now. How about this? I'm going to kind of highlight in yellow this little thing that kind of goes around, looks maybe like a skirt around this dude. That's the base plate. Anybody gather what the role of the base plate is or what it might be involved in? If not, that's okay. We'll come back to it in a minute.
Well, it looks like a point of attachment for the tail fibers and the mucus.
It kind of is, yeah. And in fact, what we find on here is there is lysozyme. Anybody recall what lysozyme does? This is a term we brought up like 4 or 5 weeks ago. Lysozyme breaks down cell walls. Okay, and that needs to be, the cell wall serves as a protective barrier. So one of the things we'll talk about is this base plate is involved in what we call the penetration step, in which the bacteriophage actually injects its genetic material into the host. And that injection occurs through this little tube that's inside of the sheath. So one of the things that's really fascinating about these bacteriophages in this process is probably the image that most of you drew is this in an elongated form. This is what we would call the contracted form. So these bacteriophage, what they will do is those tail fibers will sit on the cell, and that triggers, without any energy whatsoever, it triggers some sort of mechanical process in which that sheath there goes boom, and as that constricts, what that allows for is the pushing of this internal tube through the outer membrane. It'll, with the addition of the lysozyme, it helps it to push through the peptidoglycan and then ultimately through the inner membrane as well. The DNA then that's in that capsid, kind of by passive diffusion, will work its way out of the head and pass through this hollow tubing to get to the inside of the bacterial cell. So in the case of a bacteriophage infection, the only thing that makes its way from this structure into the host cell is the genetic material. And that's it. At that point, the virus requires some host proteins in order to support its life cycle. And this is something that's true about all viruses. They rely on the host. They can't replicate on their own. Any questions about the structure? Okay. So let's talk about how this relates to the life cycle. Bacteriophage and all viruses basically have 6 or 7 common steps to their life cycle. 1, attachment. What we find in attachment is an interaction between, in this case, it would be between the tail fibers and what we would call a receptor on the host cell. When we talk about eukaryotic viruses, instead of tail fibers, they have what we call receptor binding proteins or spikes. Okay? But they perform the same function. So that's step 1. Step 2, we can describe as entry or penetration, and the term that we use is specific to the types of viruses. Bacteriophage, we use penetration. Human eukaryotic viruses, we use entry. Honestly, if you get the two mixed up, I'm not that worried about it. Okay. The third is what we call biosynthesis. Biosynthesis encompasses replication, transcription, translation. So in this process, what we find is that the virus is somehow making more viral genomes. Sometimes that's using the proteins it encodes, sometimes that's using proteins that the host encodes. We find transcription of, in this case for bacteriophages, mRNAs, which ultimately will be translated into protein. So they follow the same central dogma here that we've talked about with prokaryotic cells. After biosynthesis, we begin to get assembly, in which the individual proteins that make up that bacteriophage structure start to just kind of come together or assemble on their own. It doesn't require energy for them to assemble. Simply, it's that they kind of fit together like puzzle pieces in some ways to help, and the high enough concentrations of them encourage them to come and clump together. At the same time, we get packaging of that genome back into that capsid. All right, so that's all assembly. We might see in some viruses an additional maturation step where they've got to undergo some additional modifications, breaking down of protein. This is not a step we will talk about. And then finally, exit, sometimes referred to as egress. But this goal here then is to get out of the cell so that they can infect more. Bacteriophage, the lytic life cycle, will go through these stages in exactly this order. And it doesn't take much time. For example, T4, which is one of the most common E. coli bacteriophages, probably the most well studied, it can get in, infect the cell, and get out in under 30 minutes. And it goes from one virus and one viral genome to 100 within that 30 minutes. And just put that into context real quick. E. coli replicates once every 30 minutes. And replication in E. coli means one cell becomes 2. E. coli can only replicate its genome one time to produce daughter cells within 30 minutes. Viruses that infect it, 100 of them in a minute, or in 30 minutes. Okay. And we'll talk a little bit about what happens in these steps. So let's take a look at, I'm going to back up here and go to this image. So this is one that I kind of put up, and I gave you guys on the slides, a blank slide to kind of fill this in as you're going through. But if we're to walk through this, what is this first step then? What are we observing in this first image here? So let's take those words that we've just used and put them to this image. What's going on in this first step? We've got attachment. So those tail fibers are attaching to the host cell. That's kind of shown here. Oops, no it's not. Oh, bother. Oh, great Google. There we go. Okay. All right, so tail fiber there. What else is happening? What's happening to that genetic material?
It's injecting it
In other words, what is this? If it's injecting that genetic material,
into
entry or penetration, exactly. So there's two steps that's occurring in this first part here. We've got attachment and we've got entry. At this point, what this is showing is this structure, this what we consider to be the structure of the bacteriophage. At this point, it detaches from the cell, and it's now just like any old protein floating around. It'll be degraded eventually, but it's of no more use for the viral life cycle. At this point, the whole life cycle is dependent on that DNA that just made it into the host cell. First thing we find, this DNA circularizes. And then we find from here to here, what steps are occurring? What's the next step in the process after penetration? Biosynthesis. So the virus, what it actually does, because it's not coming in with any proteins of its own, it actually relies on the host cell for the initiation of this process. What we find is that E. coli will, their RNA polymerase, will begin to drive transcription of the bacteriophage genome. One of the first genes it transcribes, the DNA polymerase that the virus encodes. Once that protein is translated, now the virus can begin to mass produce its own genome. And so it's going to make many, many copies of this genome in that short period of time. We also find that that RNA polymerase begins to drive transcription of genes that are required for making that viral structure. In fact, there are over 50 proteins that make up that viral structure that we looked at. And so all 50 of those proteins have to be made. In addition, we're going to find some other enzymes being produced as well that help out with some of these later steps. So we've got biosynthesis here. What's happening at this bottom image? That's assembly. The genomes make their way back into the capsid heads. Virus reforms. Somewhere in here there's maturation, and then what do we have at the end? That's our exit. And the way that bacteriophage in this lytic life cycle, which again is this one over on the left-hand side here, the way that they actually exit the cell is by producing, again, lysozyme and other lysins, break down the cell wall, and they basically burst this cell open. And so there are some really great images of bacteriophage exiting bacterial cells where it looks like basically as if you've captured a balloon in mid-pop. Or the end of that bacterial cell is now degraded, has burst, and you've got all these viral particles coming out of it. Right? Those viruses now are able to go ahead, attach to other bacterial cells in the proximity, and repeat the process all over again. Molly.
During assembly, like where do the structures that it assembles into come from?
So you mean like where do the bacteriophage structures come from? The proteins that produce those structures are all encoded for in the viral genome.
Okay, so it like builds itself back together kind of?
Pretty much, yeah. I mean, it's kind of like the cell becomes an assembly plant in some ways where from different, and we're kind of talking this in very broad strokes, but there are going to be multiple genes at different regions on the genome that are expressed. Each one is producing, it will lead to the production of a different protein that contributes to this structure here. And so this capsid is actually made up of 2 or 3 different proteins that there's a lot of them that make this structure up. What this sheath here is trying to show is that this is the same protein repeated again and again and again, and they kind of assemble in almost like a helical or a corkscrew manner. These tail fibers, there's about 6 to 8 of them on this thing. Each of those is identical. So in order to build this, what we find is that these proteins kind of they come together. They're driven by forces that we see drive the three-dimensional conformation of proteins in the first place. Hydrogen bonds, hydrophobic interactions, ionic bonds are going to help this to basically assemble out of nowhere on its own. And it's incredibly fast. And the process from about starting the transcription to the assembly of these takes about 15 minutes. So, very, very fast. So, the left-hand side represents lytic. When we typically think of an infection in a human, that's what we observe. Cold is a virus that undergoes a lytic replication. It gets in, it produces more virus, it gets out. The other side here represents, in bacteriophage, what we call lysogeny. Ah, booger. What might be analogous to this in humans would be either latent infections or potentially chronic infections, I suppose. So in this one, what we find is that the virus actually, through a mechanism that we will not discuss because it is incredibly confusing, the virus actually integrates its genome into the host genome. This is not like recombination that we saw in horizontal gene transfer, or it doesn't have to be, but the virus has enzymes that will nick itself, nick the host, and allow it to just kind of integrate into that host genome. And at this point, it goes into a quiet state. The virus is still there. It replicates every time the host replicates because its genome is now part of the host genome, but there's no viral particles being produced. What are infections that we find in humans that are analogous to this, where we have a lifelong association with the virus for the remainder of our life?
Isn't there a form of latent TB?
There is. That's kind of different, though, because TB is a bacterial infection. But there is that similar idea where there's this like lifelong association.
Like herpes simplex?
Herpes simplex is probably one of the best examples, right? We can get recurrent bouts of cold sores are the most common form or that reoccurrence that we see. Our immune system fights it off. It goes back into hiding until the next time that it's reactivated. If you've had mono, you have EBV in you still to this day. Actually, probably everybody but one person in this classroom has it because EBV infects 95% of the individuals in the world. Most of us just don't know it. Okay? Because most of us have been infected before puberty. If you're infected before puberty, there's no symptoms associated with the infection. HIV is actually probably the most analogous to this. Like lysogenic bacteriophage, HIV can integrate its genome into our genomes, particularly in T cells, the cells that it infects. And what we find in the case of bacteriophage then is that at some point after, and this number of replications there's no finite or determined amount that this can occur, but eventually what we'll find is in some forms of stress, we'll find the bacteriophage genome reactivating and going back into a lytic cycle. And what that stress is usually associated with some sort of DNA damage to the host. So usually that DNA damage to the host serves as a signal that, ah crap, this host is about to die, we better replicate and get out of here as soon as possible. That's kind of the design of this mechanism. What does this right-hand side look like with respect to what we talked about on Monday? So we talked about 3 forms of horizontal gene transfer. Which one uses bacteriophage? Transduction, yeah. And so this model, you could assume, I mean, this is analogous in some ways to transduction. Okay? So transduction can only occur if recombination can occur, or if these phage are temperate or what we call lysogenic phage, basically. All right. So let's take a look at how we can observe this infection. There's a couple of ways we can do it, and actually we can use the same mechanisms that we use to measure bacterial growth. We can do direct counts, or we can do indirect measurements. So this is what I'm showing you up here. On the left, represent two plates. The top one, basically this lightish color is all bacteria. So bacteria all over this plate. What do you notice about the one on the bottom? Okay, good. And are those spots, how would you describe those spots? You don't have to tell me what they are, but are those spots darker, lighter? Is there more bacterial growth, less bacterial growth? They are darker, and it's because they're on a black background, right? Meaning light can pass through it more, which means what about the bacterial growth there? There's less there. There's less there. Think about it in terms of inverse. So you can see better to that black surface. So these represent areas of no bacterial growth. These are what we call plaques, P-L-A-Q-U-E-S, not to be confused with dental plaque, all right? But spelled the same way. What do these plaques represent? And think about the life cycle that we just observed. What's happening at these plaques? I'll tell you, they've been infected, but the absence of bacterial growth here represents what? Yeah, who said that? Kelly? Absolutely. These cells have died. They've been killed by the bacteriophage. The bacteriophage have lysed those cells, and so there's no growth occurring there because the initial cell that was infected and the ones around it are being killed by bacteriophage. One of the things that we said is these bacteriophage, in order for their exit to occur, at this point here, they're secreting enzymes like lysins or lysozyme that will help to break down that cell wall. And in breaking down that cell wall, these cells burst to die. That's why we see these plaques form. It's because of that activity of the bacteriophage. So we can actually use this in a couple of ways. I'll explain my research for a minute. We go collect poop water, literally, from the sewerage district. We filter it to get rid of the bacteria, and then we look for bacteriophages. So we will take E. coli, mix it with the water, and we get plates that look like this. And each of these represents a different bacteriophage, each with its own unique characteristics. Okay. So this is a measurement of a way to directly count how much bacteriophage is in a sample. It's in essence the inverse of looking at a colony forming unit. But the idea is the same, that you can count a concentration of bacteriophages within a sample by counting the number of these plaques that form. So this is one way we can observe killing of bacterial cells. The other is through an indirect measurement, using spectrophotometers, something that we've all had experience with in this lab. So this graph here is one where we're looking at time in hours on the x-axis. Y-axis is optical density at 655 nanometers because the instrument we were using couldn't do 600 for us, but so be it. One of those two colors represents a culture of E. coli that's been infected also with bacteriophage. The other one represents a culture of E. coli that's been left uninfected. Take a look at the two for a minute, 30 seconds, and then turn to somebody else and tell them which one is which. Then you got to justify, of course. Which one's infected with a lytic phage and which one is not infected at all?
Oh, okay.
Yeah, so we've actually added bacteriophage to one of the two cultures.
Okay.
Well, the error bars look like that because it's on a log phase scale. We did t-tests and standard deviations. Two typical, very common microbiological. We don't believe in SPSS or other crazy nonsense. All right, so which one represents the uninfected culture? I heard somebody say the top one. Who was that? Taylor, was that you? No, Emma? Okay, you can push it on Taylor, it's okay. All right, so let's go over those phases of growth again. What's this representing here? And don't not bacteriophage growth, but regular bacterial growth. This is our, well, lag would be somewhere here. But there's our log, and then stationary by the end here, right? When we add bacteriophage, and in this particular experiment, you're going to notice first 2 hours actually, so hour 1 and hour 2 look pretty similar, right? We added bacteriophage right here at this point at 2 hours. And what happens? Some bacterial growth here, and then what happens at that after that point? So what's happening? Why does the OD go down? Okay, so in comparison to what we saw over here, those spots are letting more light through. Or another way to say this is that those bacteriophage are lysing those bacterial cells, which is dispersing their membranes. And in essence, what that does, it allows for more light to get through and doesn't impede that movement of light to the detector. So what we're observing here is another example of a lytic bacteriophage. One in which we would see them infecting and then wait, there's no bacteria for a long period of time. But what happens right at the end here? We start to see growth again. Why? Why do we at 11 hours begin to see bacterial growth all over again? Think back to things we've talked about maybe in the past week. So last week Monday's topic was ways that bacterial cells replicate. And then the second one was on DNA to RNA to protein. And the third lesson was on gene expression, and the fourth was on horizontal gene transfer. On last week, Wednesday, when we talked about replication, we talked about mistakes. What do we call those mistakes? Mistakes to the genome would be what? Mutations. How might a bacterial mutation explain this observation? Somewhere in this population, a bacterial cell spontaneously mutated. And in that mutation, no longer can they be infected by bacteriophage. And that's where we see the regrowth. So sometimes these are called bacteriophage insensitive mutants. I just call them bacteriophage resistors. But they are organisms, they are a subpopulation of the bacterial cells that in their normal replication have probably a missense mutation that now allows them to survive even though the bacteriophage is present. Most often where we find these mutations accumulating are in the receptors that the bacteriophage attach to. So by blocking that initial step of the bacteriophage life cycle, you can block more bacteriophage from being produced. Okay. We'll come back to this. Actually, this is going to be an important topic in a couple of weeks. We're going to move into a section where we're doing viruses today and next week Wednesday, doing antibiotics for a week, and then we're going to finish up with 3 days of microbial evolution. We'll see where some researchers are actually using this as a mechanism to make bacteria antibiotic sensitive. Okay, so we'll talk about that later on. But these probably represent organisms that are now completely resistant to the bacteriophage. All right, so let's finish off with this. I'm going to give you guys, I want to make sure to reserve about 3 minutes to talk about homework assignment and to talk about Wednesday. So let's take 4 minutes, and I'd like you to just go ahead and list what are characteristics of the lytic life cycle and what are characteristics of the lysogenic life cycle, based on what we've talked about today and based on any other information that you have. Take 4 minutes to work in a group on this. Again, Annie, you may not. Characteristics of either. Why don't we just start with lytic? Characteristics of the lytic life cycle. Okay, so it's a productive infection, right? Produces phage, as they're sometimes referred to. What else? Not only do they produce phage, but what happens to the host? Kills the bacterial cell. All right, there's another characteristic. Others? I honestly, I don't mind you saying that, because I think that actually the lessons we know about bacteriophage actually can inform us about human infections as well. And so, yeah, I think that's going to be a comparison that you will see. So sometimes the term we use is acute, right? But these would be ones where signs and symptoms of illness are immediately apparent. Yeah. Oops. Immediate symptoms. And believe it or not, bacterial cells do kind of start to produce proteins that are and would be signs of like, hey, I'm infected, basically. So, any others to add to that one?
It happens quickly.
Sure. Quick. And by quick, we mean minutes to maybe hours, depending on the host cell, depending on the bacteriophage. How about lysogenic? What are characteristics of the lysogenic life cycle?
It integrates its genome into the host genome.
Good. Integration. Does good. And in terms of it's dependent on the host cell for those replications, right? So maybe we can put something here as a caveat, like replication. I want to say something like without producing viral particles or something like that. Sorry, I didn't bring my pen, so the handwriting is really crappy. We'll say that Reed wrote it. One of the characteristics of this is that it can be a very long established infection, okay? And again, what triggers a cell to go from, or what triggers a bacteriophage to go from lysogenic back into lytic? Or what triggers HSV to go from latency into a lytic infection? Yeah, that's the basic term. Stress. Or in the life of bacteriophage, usually UV damage or some other damage to the genome. That's a pretty good list. Any questions on what we've talked about? Okay, so stay in your groups for just a minute. A couple of things for Wednesday. Wednesday, I'm not lecturing. Yay! So somebody from UW, grad student from there, is going to come and talk about 3 different human viruses, just small pieces of each of them. There is no mini lecture for this one. I do have some areas of the book to look at if you're interested. There is a homework assignment, but it's a very small one. So the homework assignment, between the 3 of you, one person needs to pick influenza, one person needs to pick human papillomavirus, and one person needs to pick HIV. And you'll see there's a journal entry for you to fill out. I probably should turn this into a wiki, but we're just going to leave it as a journal entry right now. Describe a sign or a symptom associated with that illness or virus, and how does the virus actually cause that to happen? So this is going to require a little bit of outside investigation on Wikipedia, basically, okay? But take the time to try and figure out one sign or symptom and what contributes to that. And between the 3 of you, you can decide who's going to do which part, okay? Lauren? It's due Wednesday, or Tuesday night. So before you come to class on Wednesday, and actually what we'll do is we'll spend the first 5 minutes or so on Wednesday getting back into these groups and talking out these signs and symptoms, okay? Any questions? I will post slides for this section in a matter of a day or 2. I just haven't yet because I'm waiting for Kathleen to update her stuff. Other than that, I have quizzes from yesterday if you want to grab them. Otherwise, I will give them to you when you are in lab tomorrow. I did as I was walking by.